Okay, folks, so it gives me great pleasure to welcome Dr. Michelle Toe from Lancaster University. Michelle started about the same time as most of you know. 50% of my job is now back at Lancaster, and Michelle and I started pretty much the same time. Michelle is a vision scientist now, but she started off, she did two BAs, two bachelor degrees rather, at McGill University, one in physiology and mathematics, and one in psychology. And then she moved to the University of Cambridge for experimental psychology, where she did an MPhil and then a PhD in experimental psychology. And she worked also for a postdoc in the department in Cambridge. And then she went from there to a lectureship at Hull University, which is probably the most glamorous of the three institutions.
Yes.
If anybody's from Hull here, I apologise. Hull's very nice. As I mentioned, Michelle is a vision scientist, but she's very much interested, as are several of us now in the group. We're sort of forming a sort of sensory or multimodal group at Lancaster in multimodal perception. And so she's going to talk a little bit about some of her vision work, I think, and then sort of talk about some of the work she's done on combining visual and auditory information. I think might be of interest to some people. So, thank you very much, Michelle.
Thank you very much for inviting me. So today I'll be talking to you about modeling feature integration in vision, audition, and across modalities. And by the end, I hope to convince you that there's one general rule that can model all these different types of feature integration. So I will start by talking about feature integration in visual stimuli, so natural scenes, natural photographs, and complex images. Then I'll talk about modeling feature integration in auditory stimuli, so I will be talking about using musical sequences and phonetic sequences. And lastly, audio-visual stimuli, where you have an image matched with an audio clip. Just a quick recap on what feature integration is for those of you who might have forgotten. If you look at the bottom, you have this lovely picture of Lancaster, where I work. And you see that there's a lot of lots of information. You have color, you have shape, you have depth, you have orientation. And if this were a movie clip, you would also have motion, okay? Now, although all this information reaches your eye, reaches your retina at the same time, your brain does not process it all at once. What it does is it processes it in more or less different pathways, okay? And we know that because you can be color deficient, but you can still manage to process depth, motion, and orientation correctly. Similarly, you can have a disorder of motion processing. You can suffer from something called akinetopsia, where you cannot process motion, but you can process color, depth, and orientation accurately. So you know that you have these more or less separate pathways. Now, after the brain processes this information separately, what it needs to do is to combine them back together again to generate your rich visual experience. And it is this process here that is called feature integration. So when the cues come back together to generate your sensory experience. And that's what I'm interested in. There have been several different models for feature integration, including linear addition. So some people say that the cues just add up together using linear addition, like just the sum. Others have suggested that it's more of a Euclidean summation, so what follows more along this type of equation. And others still suggest that it's more of the maximum rule. So the more salient of the cue takes over your sensory experience, whereas the weaker ones, they're not quite as important. Now, what's really interesting about these different types of models is that they're all examples of something called a Minkowski summation. Okay? And Minkowski summation is particularly important in vision because we tend to use it a lot when we talk about cue summation. And just to explain to you what the Minkowski summation is, to show you, this is what it is. It looks more complicated than it actually is. It's basically y equals to summing the different components of x raised to a power m, and then after you've summed them all together, you take the mth root. So if I elaborate it, I have y equals to x1 raised to the m, x2 m, x3 m, until xnm. And then after you sum all of these together, you take the mth root. Okay? So in this example, I have n elements, so it's quite a long equation. However, in my experiments, I always only have 2 components. So n is equal to 2 in my experiments, and this will be the equation that I will be referring to throughout my talk. So y is equal to x1 raised to the m plus x2 raised to the m, and then the mth root, which is equal to this. Okay, so it's quite a simple equation. I said earlier that linear addition, Euclidean summation, and maximum rules are all examples of this Minkowski summation. So what I mean is the following. So this is the Minkowski summation. If I replace the m value by 1, then I get this equation, okay, x1 to the 1, x2 to the 1, and I take the first root, and so you have just linear summation. If I replace m by 2, then I end up with Euclidean summation, which is clear here. And if I replace m by infinity, then I end up with the maximum rule. I'm not going to prove this mathematically, so you have to just believe me. Okay? So these are, so we're all very familiar with the Minkowski summation, even though we don't know the name necessarily. Okay? So in my talk, I will be talking a lot about Minkowski summation with different power values of the m. So if I say if m is equal to 2.78, this would be the equation that I would be meaning. Okay, so y would be equal to x1 raised to the 2.78 plus x2 raised to the 2.78, and then you take the 2.78 root. Okay, I'll be talking a lot about these, and we'll be seeing a lot of these. So I just wanted you to familiarize with this. Okay, so let's talk about feature integration in vision. There's many different ways you can study feature integration in vision. I'm sure a lot of you are familiar with Anne Treisman's visual search. Okay, so she talks about feature integration, and she reads, she collects reaction times. But I'm a geeky psychophysicist, so I like compound grading detection experiments. And an example of a compound grading detection experiment is the following: you present observers with a first grading, okay, and you record how sensitive they are to it by measuring the threshold, and you get the first threshold. Then what you do is you present them with a second grading, and you measure its threshold. Okay, the threshold for that grading. And what you can then do is to combine the 2 gradings, add the first grading and the second grading together. You end up with a plaid, and you record the observer's threshold for that stimulus, and you have a third threshold. Okay, and with this type of experiment, you can measure feature integration because you can see how these 2, when they sum together, how does that affect the threshold of the third stimulus, which is basically a combination of these 2. And what Robson and Graham did in the early 1980s, they found that you can predict the threshold of this grading by using a Minkowski summation, which is what I've been talking to you about, when the m value is somewhere between 3 and 4. Okay? So if you take this threshold, you raise it to a value between 3 and 4, and then you take this threshold, you raise it to a value between 3 and 4, and you take the 3 and 4/4 root, then you can generate a pretty good prediction of this threshold, okay, which is pretty cool. And this is called Minkowski summation. However cool these experiments are, there are some weaknesses. Well, first of all, when we look out in the real world, there's a lot of information. It's very rich, like this picture here. And it's not really similar to what we see. It's not similar to the gradings, which I present here on the left. Okay? So what we see in the real world is not really representative of, cannot really be representative, sorry. Gradings are not representative of what we see in the real world, okay? At the same time, experiments where you measure thresholds, they don't really represent what we see either, because when we see changes, they're quite noticeable. So when you see me talking, when you see me moving my hands, these are large, noticeable differences. They're not quite minute and quite small. So I'm interested in how people see noticeable changes in very natural stimuli. Okay? So changes that we see are big and noticeable, which is why what I've done is I've run super-threshold discrimination experiments where I present observers with pairs of complex images with large noticeable differences, and I ask them to tell me how different they appear to them. So instead of recording thresholds of gradings, I present them images with large noticeable differences. Okay? And now I'll just explain to you my experiments and I'll show you how I collect the data. So in the first experiment, I will be describing one where we investigate how cues are integrated in natural scenes. And in this one, we present observers with 900 pairs of images. They're all randomized, okay? And within these 900 image pairs, there are 136 combo sets, which I will explain to you what they are. We tested 11 naive observers, and then what we did was we compared the actual ratings of the combination change with a model prediction that we generate from the Minkowski model. So the type of stimuli I had in my experiments were something like these. So we could have things appearing, disappearing. So in this case, you have a shadow disappearing. Okay, so the shadow of this person has disappeared. You have things becoming blurry or becoming more sharp. You have color change, you have shape change, and you have texture changes. So these are quite complex changes, and they're quite noticeable apart from the texture changes. Okay? And so here are just 5 examples of the 900 that I use in my experiment. And what I did was I asked observers to rate how these differences appear to them based on something called a standard pair. And this is the standard pair that I use, and I tell people this difference is equal to 20. Okay? So this difference is equal to 20. I present to people pairs of images, and the way they rate the images is as follows. If they feel that this difference is as different as this difference, they would rate this pair 20. Okay? If they feel that this difference is less different than this difference, they would give a rating between 1 and 19. If they thought it was more different, they would give a rating of greater than 20. And if they felt that there were no differences, as in the texture example, they would give a rating of 0. Okay? And as you can imagine, it is quite difficult because these differences are across dimensions. But after a bit of practice, observers become quite good. I just tell them, don't think too much, just go with your gut feeling. Okay? And the way I presented the images are as follows. So there's a fixation point. I present first image, second fixation point, second image, fixation point, first image again. The reason why we have this setup is because the images are randomized, and we want them to be presented with both changes. Okay? And then at the end of that, there's a random number between 10 and 30, and the observers are asked to modify that number to generate the number they want to enter. The reason why the number is on the screen and is randomized is so that we don't have an anchoring effect, and the number is on the screen so that the observers don't have to move their eyes away from the screen to type on a keyboard. Okay? I mentioned combination sets when I was first describing this experiment, and here's an example of a combination set. So in the first pair, you have a change along one dimension, so there's a change in shape. And then you have a second pair where you have a change in a second dimension, change in color, okay? And then you have a third pair where you have a change in shape and color. And observers would give this a third rating, okay? And the question is, if you have the observers' ratings for R1, and you know what they rated the pair for R2, can you predict what would R3 be, okay? And here are just other examples of combination sets, just to show you that there's quite a lot of variety. In this case, it's a hue change. In this case, it's a flower disappearing. And then in this case, it's a hue change, or a flower appearing, and a flower appearing. Okay? And in this case, shape change, image becomes blurry, and image becoming blurry, and a shape change. So how are the cues combined? So is R3 just a linear sum of R1 and R2? Or is R3 the maximum of R1 and R2? Or could it be that R3 is something similar to what Robson and Graham found? So using a Minkowski summation with a power between 3 and 4, can you predict R3? So we decided we want to look at that. So here I have the first set of results. On the x-axis, I have the actual value of the measured R3, so what people entered as R3. On the y-axis, I have the linear summation. So if I add R1 and R2, this is the value I would get. This line is called the identity line. So had the predictions been perfect and the same as the measured R3, all the red dots should be around this line, okay? But they're not, which suggests that if you take a linear summation of R1 and R2, you tend to overestimate the actual prediction, or the actual measurement, okay? So if you just take this value, this rating, and this rating, and then you tend to overestimate what people actually rated for the R3. The second model I looked at was the maximum rule. So what if you took the largest of R1 and R2? In this case, you see, although the dots are closer to the line, they tend to be a bit under. So by taking the maximum, you tend to underestimate the actual values of R3. Okay. So to find what is the actual best fitting value of M, what I did was I just ran an iterative fit. Okay. So if you look here at the x-axis, you have different values of M. Okay, so I ran the model for different values of M, and on the y-axis, I have the sum of squares. So that's basically the error, the difference between the measured and the predicted. Okay, so because this is a sum of squares, the higher the value, the less accurate your model. The lower value, the more accurate your model. So you want the dots to be closest to 0 as possible. That tells you which values are the best ones. And when you do this type of fit, you find that the best M value to predict the actual R3 is 2.78. Okay, so the best model for predicting R3 is a Minkowski with a power of 2.78. And just to demonstrate to you with actual numbers and examples, so here are 4 examples of combination sets. We have R1, R2, R3. Okay, here are the actual ratings. Okay, these average A ratings of like 11 observers. And I just mentioned that this Minkowski with M value of 2.78 generated the most accurate predictions. And if I actually calculated estimates, these are the values. Okay, and if you compare them, 28.6 and 27.82, 24.26, 25, you can see that they're actually quite close. Now, here I present them all together, but I'd just like to remind you that we presented 900 pairs of images, and they were all randomized. Okay. In addition, if you remember how difficult it is to rate these images, there's actually quite a lot of subjectivity. And the fact that we could still manage to get quite accurate predictions is pretty fantastic. Okay, so I was really, really excited about that. So just a summary. If you look at linear summation, you tend to over-predict R3. If you look at maximum, it tends to under-predict. But when you use the Minkowski summation with M equals to 2.78, you get pretty good estimations, which is pretty cool. So in the second experiment, we noticed that when you have the content of the images mattered. So if you have a smiley face versus a slightly frowny face, observers tend to rate it quite high, whereas if you have a shadow disappearing, they might rate it quite low because it doesn't really matter the difference. So the second experiment, we want to eliminate the high-level information and just really look at how the low-level cues combine. Okay? So the way I did that was to alter the images. I took the original image, I distorted the pixels, and I inverted it just to remove the content, just to make it more difficult for observers to see what the contents were. And in this case, these are just distorted versions of the previous ones that I've shown you. And as you can see, it just becomes more difficult to see what you're actually looking at, okay, identifying it. But we use the same standard pair. And in this experiment, a combination set would look like this, okay? So we have the same number of combination sets. And the question again is, can you predict R3 from R1 and R2? And you find that, yes, you can, okay? With a Minkowski power of M.279. So I used the same type of analysis, I ran the same type of analysis, and I found that linear summation tends to overestimate, maximum rule tends to underestimate, but if you use a Minkowski summation with M.2.79, then it generates the best predictions. Okay. So I ran a third vision experiment, because in the first two we had random types of changes, like with random magnitudes. So you could have a bright color change or small color change, and you could have shade changes. But I wanted to do this more systematically. So in this case, what I did was I used synthetic stimuli, where I could alter something like bush size. Okay, so this is the original image. I can make the bushes smaller or larger. Okay, and along a different dimension, I can alter the shadow contrast. So the shadow contrast is very high here, and it's much lower there. Okay, so in this case, I have a very systematic set of stimuli. And in this experiment, we had 432 combination sets. And when I ran the analysis, the best estimations were obtained when the Minkowski value is 2.95, which is quite similar to 2.78 and 2.79 that we found previously. Okay? Even though this is a completely different set of observers, different set of stimuli. Okay? And again, linear summation overestimates. Maximum does a pretty good job, it tends to underestimate, and it's really the Minkowski summation that generates the best predictions. So here's the summary, and as you can see here, the M value is always close to 3, so it's between 2.5 and 3. Okay, which is very interesting because it is consistent. And I also present you here the correlation values, which are quite high. Then what I did was I just combined all the combination sets from all 3 experiments. And when you do that, the best power is, again, close to 3. It's 2.84. And here we have 704 combination sets. So, and when you look at the correlation value between predicted and actual, it's 0.96. Okay? So, the vision experiments seem to show that a Minkowski summation with power close to 3 can be extended to feature integration using complex natural images, but not just small changes, but with large changes. And this is very interesting because as a post-grad, when I was a PhD student, I always learned that, if you want to be really, really methodical, you have to use gratings, and everything has to be very well controlled. And this experiment shows you that even when you don't use gratings, that you can find the same type of findings. So it's quite nice to know that your little grading experiments can be expanded to real, I guess, more realistic stimuli. Okay, so this is similar to what Watson and Graham found. Then I thought, well, so now we know about feature integration in vision. What about feature integration in hearing? And I'm a bit scared because you guys are the experts in hearing, and I'm not. Okay, we know that feature integration in hearing is slightly different than vision. But I read a paper from Green that suggested that the detectability, so the thresholds for detecting a compound stimulus, is always a bit better than when you compare it with the thresholds for the individual ones. So maybe there is some type of summation going on, okay, a nonlinear summation. And around this time, when I was thinking of ideas for experiments, my husband was running, the first language and music conference in Cambridge. So then I thought, oh, I could do an experiment with language, phonetic, and I can do an experiment with music. So there you go. So these are the next 2 experiments I'll be describing to you. Okay. So in experiment 4, I looked at how cues are integrated in musical sequences. And in this case, we have 96 combination sets, 15 naive observers, and I use the same type of analysis. And to wake you up, I have some musical stimuli to show you, so you can listen to. In this case, we have a standard pair, so I use musical sequences, and this is the standard. So I tell listeners or observers, this difference is equal to 20. Okay? Can you hear? Present them with various types of differences. So there could be changes in dynamics. Okay. Could be changes in notes or articulations. Okay. Transpositions. And then changes in instrument or changes in tempo. Okay. So again, it is quite difficult, but with a bit of training, they can generate quite reliable ratings. And in this case, a combination set would be something like this. So in a first pair, you would have change in dynamics. In a second pair, you can have transposition. And in the third pair, you have both. Okay, and you want to see, can this rating and this rating predict the third one? Same type of analysis, and when you do that, and in this case, I also looked at Euclidean summation. I found that the best predictions were always obtained when m is equal to close to 3. So in this case, it's 2.95. So again, linear summation overestimates, Euclidean overestimates a bit, maximum tends to underestimate, and Minkowski does a pretty good job when m is equal to 2.95. Okay. I also ran a post-op test just to see whether there are any differences in the different types of combination, and there were no differences. So the Minkowski with m equals to 2.95 generated the best predictions across the different types of combination. Okay. Then in the fifth experiment, I looked at phonetic utterances. So instead of musical sequences, I just had people or myself pronounce vowel sounds. Okay, and I modified how they sounded. And here are some examples. Okay, so this is the standard pair.
Da.
Da.
Okay, so that difference is 20. And then I presented different types of changes. So intensity.
B. B.
Okay. Pitch.
Ko. Ko.
Okay. Tempo.
Lu. Lu.
So that was just slightly faster. Spectrum.
Ra. Ra.
So I removed some of the frequencies. And the syllable.
Yi. Ya.
So that's completely different. Okay? And an example of combination set in this experiment would be something like this.
Gu. Gu.
Intensity change.
Gu. So.
Syllable change.
Gu. So.
Okay, and it's the same type of analysis. So how does R1 and R2, how do they need to be combined in order to generate the best predictions? And in this case, it's with Minkowski value of 2.54. So it's consistently between 2.5 and 3, which is quite cool. Okay, again, linear summation overestimates, Euclidean overestimates a bit, maximum underestimates, and Minkowski 2.5 generates the best results. And again, I looked at the post hoc. I did a post hoc analysis to see whether the different combination types are, I guess, the estimates, whether they're different for the different types of combination, and they're not. So it's consistently good for all different types of integration across dimensions. So the auditory experiments show that a Minkowski summation with power between 2.5 and 3 can be extended to feature integration in natural auditory stimuli. Now, having done vision and audition, the next natural step is to do audiovisual. So what I did was I used images and I matched them with audio clips. So as the audio clip was playing, there would be an image, and when the audio clip ended, the image would disappear. Okay. In this situation, I also tried to test incongruities. Okay. So I had 4 categories of stimuli. I had animals, instruments, objects, and people. And the images could change along certain dimensions, and the sound could change in other dimensions. And I had 324 consistent and 324 inconsistent pairs, which I will show you what I mean by that. And in this experiment, this is the standard pair. So people will see the image when they hear the sound, but when the sound disappears, the image disappears as well. Okay. And I asked observers to rate the whole sensory experience, so not just the audition or not just the visual. Okay, it's quite difficult. And a combination set would be something like this in this experiment. So in the first case, only the image changes. Okay, so the sound remains the same. Okay. In the second case, only the sound changes, but the image remains the same. And in the third case, we have both an image change and a sound change. And we want to see, can x1 and x2 predict y? And I mentioned that we have congruous and incongruous conditions. Yeah. So in the congruous condition, you had a bird sound with a bird. Okay. But in the incongruous conditions, you could have photo of a lady sneezing and then bird sound. Okay. Or a bird image with a telephone sound. Okay. And I wanted to see whether the Minkowski value was related to the stimulus, or whether it's more of a neural thing. Okay, whether it's stimulus specific or neural. And if it were neural, then you would have the same Minkowski value for all. But if it was stimulus specific, you might have something different if it's congruous versus incongruous. That was the logic behind that design. So the congruous results, best predictions were obtained with a Minkowski of 2.62. Okay, again, in the same range. And for the incongruous combination sets, best predictions were obtained with a Minkowski of 2.5. Okay, so when you use the sum, the Euclidean you tend to overestimate, and again, maximum tends to underestimate. So the cross-modal experiments show that the same kind of Minkowski summation with a similar m value could be used to generate pretty good estimations of of R3, which is how the features integrate. And the absence of an effect of congruency suggests that it's a general neural property rather than a stimulus-specific phenomenon. So I've shown you today that Minkowski summation with a power of between 2.5 and 3 can model feature integration of natural scenes, music, language, and audiovisual stimuli. And so I like to think that there's one rule to bind them all. Which is quite geeky. I'm sure you guys are all wondering now, well, what does this m value 2.5 to 3 mean? Because I keep talking about it, but what does it mean? Well, I think, or me and my collaborators think that maybe the exponent relates to the degree of correlation of activities between the different neurons in our sensory system when they're being stimulated by natural stimuli. Now let me explain what I mean by that, okay? So suppose the activity between cells is completely independent. So the cells process information that is completely different from one another, okay? And each cell gives you a uniquely important information. So when you have a change in color, so only cell X would fire, okay? And when you have a change in shape, only Y would fire, okay? So if you have a change in color and shape, X and Y will both fire, in which case you have linear summation. M would be equal to 1. If instead of that, you have, so instead of X firing or not firing when there's a change in color, if X fires more and more when there's a change in color, and Y fires continuously more when there's a change in shape, then when you have a change in color and shape, the Z would be it would follow something more like a Pythagoras rule or a Euclidean summation, in which case you would have the Euclidean summation. And in this case, X and Y are also independent because they're orthogonal from one another. Okay? So if they're independent, you would either expect an M value of 1 or M value of 2. Now, if the cell responses are highly correlated, which means that the information given by only one neuron is sufficient, and they always do the same thing, okay, this is what you would expect. If there's a change in color, X and Y will fire, both, okay, because they do the same thing, and there's a change. And if there's a change in shape, again, they do the same thing, they just get excited and all happy. And when you have a change in color and shape, then all you need is the larger of the two, because they do the same thing anyways, so only the maximum would be important, okay? And in this case, the M value is equal to infinity, because it's the maximum rule. Now, how much correlation between cells would an M value of 2.5 to 3 suggest? In order to determine that, what I did was I looked at a different equation called the Mahalanobis sum. It's basically an adjusted Euclidean measure of similarity. So it's like the Euclidean measure, but it takes into account that the different dimensions may not be completely independent, okay? And what it does is it has this parameter here called rho, and rho represents the correlation between the dimensions represented by R1 and R2. So So this is how correlated R1 and R2 are. If R1 and R2 are not correlated, rho would be 0, in which case you just end up with Euclidean summation. Okay, so if you replace rho by 0, then you have Euclidean summation. But here it just allows you the possibility that the two dimensions may not be completely uncorrelated. So how much rho does my data suggest? I ran a similar type of iterative fit. So I look at, on the x-axis, I looked at different values of rho, different values of correlation, and on the y-axis, again, I look at the sum of squares. So what is the difference between predicted and actual measurements? And again, in this case, you want the SSQs to be the lowest possible, which suggests the lowest error rate possible. And from the vision experiments, you see that the data suggests that there would be a correlation between 0.1 and 0.2. Okay? If I did this for the music experiment, it's between 0.15 and 0.25. Phonetics is between 0.05 and 0.15. And in the cross-modal experiment, it's between 0.1 and 0.2. Okay? So it's always around this region. So these experiments seem to suggest that whenever we look at feature integration, the features seem to correlate a bit. Okay? Now, does correlation of 0.1 to 0.2 match physiological data? So if you actually took a cat, or I don't think you took a human, if you take a cat and you present it with natural stimuli and you measured its brain activity, you measured the activity of the cells, how would the cells correlate if you present them with color change versus shape change? And what we find that yes, it agrees with electrophysiological findings with a signal correlations between 0.1 and 0.2. And there's like a large body of reports, of your papers citing this range of correlation. So, conclusion, there seems to exist a general rule for feature integration in vision, audition, and across modalities. It's not the linear city block rule or the Euclidean summation or maximum rules that have been so popular, but it is a Minkowski summation with a power of 2.5 to 3, and it's that because the Minkowski summation reflects a degree of correlation between the neural signals. That's why we think it's consistent. And I'd like to thank David Torres, who is my postdoc advisor, Roland Badley, who proposed using the Mahalanobis sum, and the late Tom Trushenko, who is also my postdoc advisor. Thank you.
Great, thank you very much. It's very interesting. Would anybody like to jump in the question? I have one just to get us going, if you don't mind. You talked about at the end, and this is, of course, is very interesting to me, about how neurons process these complex stimuli, and about the sort of correlation between different neurons. So you're talking there effectively, I mean, for the neural response to correlate, essentially there has to be some sort of connectivity between them. Now, just sort of thinking about this, it seems to me that this would vary depending on where in the visual and auditory pathways, say, these neurons are located. Do you think, a neuron in the auditory nerve,
yes,
for example, is taking information directly from the cochlea, or the neuron in the optic nerve? Those two are going to be, fairly uncorrelated. I mean, you'd have thought, right? Yes. So that if you have a sound, the auditory neurons will respond, the visual isn't, and vice versa. So when you're talking about correlation between neurons, at what level of representation are you talking about?
I think it's probably the higher level.
I mean, the measurements that were made, what sort of regions of the brain were they studying?
I would think it's when all the information gets combined across the modalities. So it's quite a high level, I think. And I don't think I've explained this clearly, but one of the reasons why the activity of the neurons would be important and why the ratings reflect the activity of the neurons is that we assume that the more active your neurons, the more you will notice a difference, so the higher your ratings would be, which I'll probably skip that step.
Okay.
Yeah. So I think it's where all the information gets integrated, like audio-visual, or all the senses.
So these are neurons that are responding primarily to one sense or another, right? In the obviously in the cross-modal thing, but they're in a region of the brain still which is involved integrating. Is that kind
I think so.
That's kind of your
That's what I.
So they're separate but being processed in one group. So the kind of association cortex.
Yes. Because even when there's just an image change or an auditory change, you still have the visual that remains the same, or the auditory that remains the same. If you remember.
Yeah, sure.
So you still have the visual information.
Yeah,
auditory.
sure.
I could run one where there's only vision and then there's only audition,
Yeah.
and then combining the two. In which case you might have a different cast.
Right, yeah, okay.
So I only present the visual
Yeah,
change.
yeah,
no
I
audition.
understand, yeah. I wonder if that would still work.
Student project.
Yeah.
So looking at all this for the first time, Michelle, I can understand that Mikowski improves things, but actually the maximum one's not very far away from most of this either. In the first order of magnitude, for a lay person like me, where you say you can change two things and then put them together, it's the one thing that changed the most is most likely to predict. That seems to be what happens. And then you can tweak it a little bit with your modelling. Except if I got it right, when you did the cross-modal stuff, that was when the maximum started to break down more, is it? Because all along I was thinking, I just need to remember the maximum one is kind of close and it's not perfect, but then this one really started to look like breaking down. Yeah, that's right.
Yeah.
But, so you might have told us, it might be a bit slow, but why do you think it's poorer then with the cross-modal versus the two changes within one sense or modality?
Can you repeat the last question?
So I'm just wondering,
Yes.
it might not even be a sensible question, you can correct me. It's not. It breaks down more here. So why should it break down more with cross-modal versus when you have the two changes within one modality?
Yeah. It might have to do with what you just said. If it's within the same modality, maybe like the larger one is just the one that takes over. But if you move to the associative areas, maybe it's something different. Does that make sense?
Yeah.
Oh, I think about it over sandwiches, but
Yeah. Yeah.
you'll be adding smell next to all this, will you?
It's complicated with smell because it's difficult to control.
Probably a cat. I imagine. Yeah, I was thinking you're like a cow smelling of a rose and a rose smelling of a cow.
So you've got a question over here.
Yeah.
Can you be certain that integration is actually occurring? Because I'm thinking in terms of your bird with like a random sound. Normally when you're looking at integration, I'm thinking in terms of like the McGurk effect and that sort of thing, you've actually got movement.
Yes.
And you're seeing a picture of the mouth moving with a sound
Yes.
which changes. So, I mean, would it be possible to do this sort of thing with like a real multi-sensory scene rather than two quite disjointed scenes? Because obviously a bird moving up and down and changing its in its bird song is quite different and quite
Yes.
two different contexts.
That is a project that I ran with a student last year, and it's a flawed project. That is something that I'm doing at the moment. Yes. Also, another shortcoming of the cross-modal experiment is sometimes I would have like an animal, and there would be music or an animal coughing, suppose, and the subjects would say that, well, during the briefing they would say, well, I know it's not supposed to be congruent, but I see so many cartoons and television does so many different weird things, so I know it's incongruent, but it doesn't really feel that incongruent and change. So if you do something like McGurk, definitely, that would be a better way.
Do you think, so getting back to your point, that it's not quite strong, do you think that would be because integration relies on two weaker stimuli, whereas because you've got two supra threshold stimuli here, that's why you're not getting quite as much integration, so quite as much power effect?
It is possible, yes.
Michelle, so have you thought about whether there's any sort of hierarchy between the emphasis that people might put on certain cues? So I'm thinking in the visual instance, the reference was a change in colour. So is that in some way, you said you wanted to avoid an anchor point, but if actually colour is, because then on some of the ratings, think the average rating for one of the colour changes was 14, which suggested they didn't think those changes were as strong as the standard,
Yes.
even though the change was the same. So I'm just wondering if you used a different reference. So if you had a blurry image and said that's your 20 mark, would that change the way that they're perceived to be? Is it that some of them are more salient than others, which is what you're getting at?
Well, we ran a pilot where we asked people just to rate loads of images, and we found that the flower pair was the one that was in the middle. So that's how we picked it. But it is possible that if we pick something like a blur change or a texture change, then that could just, just magnify or diminish all the ratings.
It's more sort of comment and just something to think about, really. But obviously this group is largely composed of people interested in audiology and problems of the ear. But a lot of the things that we have to think about in when we're trying to understand, so the sort of problems faced by someone with hearing impairment is how they use the visual environment and how they might integrate that information. And I was thinking particularly the question there about sort of the McGurk effect and the lip reading and things like that. I mean, there are, I haven't really started sort of this sort of process of thinking about the sort of experiments you might do, but it might be interesting to see how people combine that sort of information and whether it's consistent with what you find about other types of information, and what the impact of, say, visual impairment and or hearing impairment are on your ability to use these different cues. You see what I mean? And you could potentially use this type of analysis to build some sort of model of how the different information is going to be combined in different circumstances that could give you some sort of predictive power, so how well someone will understand speech given this certain type of degraded or whatever information. You see what I'm getting at here?
Trying
to give. I'm just trying to sort of put some sort of clinical sort of bent on it. This is one of the things that sort of occurred to me when you were talking about it. It might be kind of interesting if we wanted to go down the sort of clinical sort of MRC grant route. We could think about doing something along those lines. Maybe. Great, okay, right, time for food. So thank you very much again, Michelle. It's a great talk and great to have you here, and look forward to catching up later, I hope. Okay, right.